Hi, I'm back for my second lecture now. And this is going to be quite different from the first one you heard. First one was the science that goes on in my lab. This talk is why I do that science and why we all have to be aware of what's going on in this global village that we live in. It has become clear that global infectious disease threat is real and something we have to worry about. Today, infectious diseases are the leading cause of death worldwide and the third leading cause of death in the United States. Now, the diseases that predominate, we all know about and have heard about. Of course, they include HIV/AIDS, they include acute lower respiratory infections, diarrheal diseases, tuberculosis, malaria. But since 1973, at least 29 previously unknown diseases have emerged. Now, 20 diseases have reemerged in new places where they never were before and are sitting in new ecosystems. And the question, of course, is why is this happening? Where do new diseases come from? Why are we facing such an escalation? And in point of fact, this is due to the very dramatic changes in the society and in the environment in which we live. There's been an explosion in population growth, spreading poverty, global warming, and urban migration. And what we're finding is new pathogens in new places and old pathogens in new places. And what do we mean by how is this happening? So if we have urban migration, which means we're moving into our forests, a typical disease that is now blooming all over the place is Lyme disease, carried by a tick. And that was due to deforestation to make way for new homes. And this causes a tick bite that's carrying this bacterial infection is quite devastating. Another, of course, that we all know about is HIV/AIDS, that is growing and spreading in all urban populations. Another interesting one is called hantavirus. This virus was unknown. It's new to us, and it appears in the American far west, in Utah and Arizona. It's carried by mice and rats. This is a very nasty virus with a 60% kill rate, and we're still trying to understand it. Another one that's coming out of the forests in Africa is caused by a virus, and it's Ebola. And that is a very difficult virus to deal with. Now, we have also the different kinds of changes in our environment. For example, mad cow disease, which you've all read about and heard about, which is caused not by a bacterium, not by a virus, but by a protein that changes conformation and goes into a state that causes a very severe neurological brain defect. And why did this somehow get out of the box? And if you remember, in England several years ago, there was just an outbreak of mad cow disease. And it turned out to be due to the fact that the production of the foodstuffs that we feed all of our livestock was made differently. We were feeding them various kinds of vegetable, mineral, and animal refuse. And this time they would include the nervous structure, the brains and the nerves. And that's where these prions were. And then it wasn't until the mandate came down to change the preparation of food for our livestock that this epidemic was dropped down. And this had to be changed worldwide. So this is another example of us trying to survive in large populations and feed everybody by changing the way in which we carry out what we do. The other thing that's happened is that there's been a resurgence of very drug-resistant tuberculosis. And in South Africa now, there are strains of the bacillus that causes tuberculosis that are resistant to every known antibiotic. And it's a particularly virulent strain with a very high kill rate. So the crowding, urban mixing of people and pathogens has given us a bloom of bacteria that are resistant to antibiotics. Now, we also know that we have international travel everywhere. It's increased. If we have a disease outbreak in Kuala Lumpur, in a day, the person who gets on that airplane with an infectious disease can be in Chicago in that same day. So that we are rapidly moving all over this globe, and there is an incredibly rapid spread of disease, reminding us that no country is an island, and we now live in a global village, which means that all the countries on this globe have to now coordinate, collaborate, and help one another to identify diseases and to disseminate things that will help squash down an outbreak of some kind. Now, one other thing that happens when this guy picks up a disease in Kuala Lumpur and winds up in Chicago. Now, the people in Kuala Lumpur may have been living with this disease for a little while and they've built up immunity. But the people in Chicago have no immunity to this, and so it's a bigger problem. Now, the part of this that's difficult is that we have asymptomatic travel. You get on a plane and you feel fine, but in fact you're infected. And one of the big problems with a possible influenza epidemic is if you catch a flu bug, influenza, you are asymptomatic for at least 2 days. And during that period of time, you are infectious. So you don't know what you're transferring. And that is what leads you to pandemic. Now, another problem, of course, is the loss of control of our national borders. And we're really not very good at carrying out our quarantine laws. It's difficult to do this. And we are going to return to this later in my talk, because in many diseases, effective quarantine laws are the only thing that we're going to use to protect ourselves. Now, every one of these issues is something that is being worked on and understood by many countries, coordinated by the World Health Organization. The final issue that is almost making where we live now a perfect storm is that while we have increased globalization, while we have increased population, while we have increased urbanization and the migration of pathogens into new ecological niches, we have the rise of antibiotic-resistant pathogens. So let me tell you what an antibiotic is. An antibiotic is a small compound, either made in the laboratory or made by some living creature. And when this target is made, when this compound is made, what it does is kill the pathogen, kills the bacterial pathogen. Perhaps I should just remind you of the difference between a bacterial pathogen and a viral pathogen. A virus is not a living cell. A virus just has a protein coat, and the genetic material sits inside this coat, and it cannot make more of itself. The only way it can make more of itself if it infects a host cell, one of our cells, one of the cells in a rat or a monkey or some other animal. And then it gets in there, co-opts the machinery of that cell, and makes many more of itself. That's a virus. A bacterial cell is this little tiny living cell that can grow and divide and respond to its environment and figure out, if it's a pathogen, how to get into a host cell and make you very ill. So that's a bacterial cell. Antibiotics are specific for bacterial cells, not viral infections. And what has happened is that we have had a history of various kinds of antibiotics, which were first discovered in 1946 with penicillin. Then soon after that, we had strep and staph infections that would be very sensitive to penicillin. Today, 80% of all strains of Staphylococcus, are resistant to penicillin. This was quite a shock to us. 1950, we had more antibiotics that would infect multiple bugs, streptomycin, chloramphenicol, tetracycline. Then in 1953, there was a Shigella outbreak in Japan, and it resulted in the appearance of a strain of dysentery bacillus that was resistant not just to one antibiotic, but to many. And that was a red flag, and people started becoming somewhat concerned. Up now to 1982, when we had the last new class of antibiotics, the quinolones, resistance is rising really in a frightening manner. Cipro, to which we were eating like candy when there was an anthrax scare in the United States, has caused enormous resistance to that particular drug. 1998, vancomycin, which is considered by many an antibiotic of last resort for staph infections and other kinds of pathogens, we are now seeing the emergence of resistance to vancomycin as well. Now, what happens when something becomes resistant to an antibiotic is it turns out that these bugs are very, very smart. And what they have learned how to do is if they see a drug coming towards it, like erythromycin, it figures out how to spit it out. Or if the antibiotic manages to get into the bug, the bacteria have figured out a way of chemically modifying that antibiotic so it's no longer working. And they also have figured out how to change the target of that antibiotic in that particular cell so it doesn't work any longer. So these bugs are very smart, and we're in a war with them, and the bugs are winning. And what we need is to understand how to make new and better antibiotics. If I just look at staph infections in the United States, in 1957, 100% sensitive to penicillin. 1982, fewer than 10% of all staph cases could be cured by penicillin. 1993, only vancomycin survived as an effective antibiotic. And today, as I've told you, there are strains that are resistant to everything. So one of the questions is, why is antibiotic resistance growing so rapidly? And in fact, what we're seeing is that antibiotics are put into animal feed, into aerosols for fruits and vegetables. Of the 50 million pounds of antibiotics produced annually in the US, 40% go into livestock. So how does this resistance arise? Let's say you have 10 to the 9th bacterial cells, all resistant to a particular antibiotic. One of those 10 to the 9th cells has a mutation that makes it resistant. All the others will be killed by that antibiotic, but that one will happily grow and divide, and then you have a bloom of an antibiotic-resistant pathogen. By feeding antibiotics in huge quantities to all of our livestock, we are increasing the chances of that one guy to develop antibiotic resistance. Another reason that things are growing, that this resistance is growing so rapidly, is that there are growing numbers of immunocompromised people. We have, and this is really partly due to the wonders of medicine and also to new infectious diseases. Chemotherapy patients have very, very low immunity. They're infected by many different bacteria, and they grow and divide and develop resistance. Transplant patients, AIDS patients, even just aging populations. If you're over 65, your immune system, is going to hell in a handbasket. And so you have to really realize that you are particularly sensitive to bacterial infections, and again, you become a reservoir for increased antibiotic resistance. There's also the excessive use of antibiotics over prescription, and then unregulated over-the-counter sales. So these are all very serious problems. Of course, as I told you before, international travel has us all over the place. And so if you get a multi-drug resistant strain of streptomycin in, Spain, you wind up with it in South Africa in 4 days if somebody is traveling rapidly. So we have complete and rapid dissemination. Now, what I'm going to turn to is the story of where new bugs come from? Where do new pathogens come from? And the story I'm going to tell you is one of E. coli 0157. It's a new and pervasive pathogen. It's a food contaminant that is now the leading cause of kidney failure in children. Now, the first time I told this story was in a very unusual scenario. It was during Bill Clinton's administration, and he became very worried about genetic engineering. In other words, what we can do in the lab now in building new groups of genes and perhaps altering a pathogen or altering some other normal process. And he was worried. He wanted to know how worried we should be about malevolent forces actually creating new pathogens. And, you know, he wanted to understand what was happening. And I was part of a group of 6 people who were invited to speak with President Clinton and his whole cabinet. And the story I told them really was that genetic engineering, yes, we can do in the lab, but the bugs and the various kinds of critters out there in the natural world are much better genetic engineers than we are, and the example is E. coli 0157. Now, this is a picture of a virus, and I'm going to show you and tell you how this figured in to the genetic engineering that was carried out by E. coli 0157. So where did it come from? E. coli 0157 was first isolated from a 50-year-old woman in California who came down with severe gastric distress and bloody diarrhea. She survived, but she was quite ill. Then in 1980, 14 children were admitted to a Toronto hospital with the same symptoms. Of these, 2 children died, and the rest were left with severe kidney damage. Again, the bug, the bacterium isolated from one of these kids was the same as that found in that 50-year-old woman. And upon analysis, the very surprising development was that this E. coli cell, which is a bug that grows in all of our gastric system and is quite harmless, had picked up a gene, a particular gene from another bug, a pathogen called Shigella, that coded for a toxin. So now we had taken an E. coli cell and put in a gene that made it a pathogen. That is genetic engineering. In 1981 in White City, California, 12 people eating in a local hamburger place became ill with the same symptoms. 1982 in Michigan, again a local hamburger place, E. coli 0157, as it was then named, was found in its meat patties. 1993, Jack in the Box restaurants in the Northwest. Hundreds became ill, 4 kids died. And this continues on and on. It was found in 1996 in contaminated apple juice and lettuce. And that turned out because the E. coli cell picked up not only a gene for the toxin, but a gene that makes it resistant to acid. So it could grow in an acid environment, which normal E. coli does not. 1997, there was again a huge recall of contaminated hamburger meat. And in 2007, just a couple of months ago, contaminated spinach was found. And that came from the runoff from livestock, which were not too far away. So it turns out that now there are 25 to 30 outbreaks per year in the United States alone of E. coli 0157 contamination, and 5% of our dairy cows carry this pathogen. So how did this happen? How do we think that this occurred? So what I'm showing you here is a bacterial virus. Remember, I told you that a virus has a protein head, and that's shown here. That's the head. This is its tail, and there's DNA in this head, and this over here shows you what the virus looks like. This over here shows you a diagram of this virus. It looks like a moon lander, doesn't it? And what this moon lander does is it lands on a bacterial cell and it injects the DNA, the genetic material, right into the bacterial cell that it hits. And this is how we believe this happened, okay? So in this diagram, I show you a Shigella bacterial cell. The blue circle indicates the chromosome, the single chromosome, and the little moon lander up there indicates the virus. So the virus injects its DNA, and that's that little circle in the center of the head, into the cell. Once that DNA gets into the cell, it codes for things that chop up that blue chromosome. You chop up that blue chromosome, and the little piece that contains that Shigella gene then gets put into the head of a new virus. And so this new bacterial virus contains its own DNA and a little bit of that Shigella DNA, and that little bit contains the gene that causes a toxin. Now what happens is that that same virus hits an E. coli cell. And we believe this happened during an epidemic of dysentery in Central America, when both E. coli and Shigella were mixed. And this virus injected its DNA into the harmless E. coli, and this piece of DNA got incorporated into the DNA of this E. coli, creating E. coli 0157. That, folks, is genetic engineering. That happens naturally. So now let me tell you a second story. And the second story deals with why it's so difficult when we're first faced with something that we haven't seen before in a given country to decide whether this is a natural outbreak, is it a malevolent deed, where is it coming from? And this is an interesting story. It turned out, now West Nile virus is an encephalitis, causes an encephalitis type disease, but it had never been found in the Western Hemisphere, okay? And this is several years ago now. And the way it started was that birds in the Bronx Zoo started to die, and they had an encephalitis type infection. And a vet in the Bronx Zoo sent her tissue samples to the CDC, Center for Disease Control, and they were pretty overwhelmed because the CDC never has enough money to do everything they have to do. And they sort of said, yeah, we'll get to this. You know, birds are dying. Well, at the same time, there was an increasing number of human patients in New York City which were exhibiting and dying of an encephalitis type disease. People thought it was the mosquito-borne St. Louis encephalitis virus. They didn't really know. And the then chief of emergency management in New York City managed to co-opt the entire supply of OFF. OFF is something that kills mosquitoes and flying bugs. And he just sprayed OFF all over the city. And he stopped the epidemic cold. Meanwhile, the lady vet at the Bronx Zoo was still trying desperately to find out why her birds were dying, waiting to hear something from the CDC, and the weeks were going on. And she happened to go to a wedding on the West Coast. And sitting next to her at this wedding was a virologist, and not particularly interested in dancing. They started talking about, you know, this odd thing that was happening to her birds at the zoo. And he said, look, why don't you send me some of your tissues and I'll try to figure out what you've got. And that's what happened. He very rapidly identified this as West Nile virus. Now, everyone's initial reaction was this couldn't be. We don't have West Nile virus in the Western Hemisphere. But at about that time in Fort Collins, the CDC had in fact identified this as well as as West Nile virus. It just took too long. And this was our first experience with trying to rapidly identify something new. Now, interestingly, concurrently, while all this was going on, an Iraqi defector had reported that Saddam Hussein was developing a strain of West Nile virus as a biological warfare agent and was preparing to release it. This was never confirmed. Was this a BW event? We don't know. Did this come into the United States on a 747 that a mosquito happened to crawl into? We don't know. We don't know the answer. But what's important and what one has to remember is anything we do to identify a new outbreak will be relevant to no matter what the source is, malevolent or natural. The problem is to understand what we've got and to rapidly understand how we can analyze these things and identify the agents. Now, this is changing, and this is changing particularly because of what we found with SARS. Now, that's happened fairly recently. SARS is severe acute respiratory syndrome. It's caused by the coronavirus, which is an RNA virus that it's similar to the viruses that cause the common cold, has a very high potential for natural evolution, so it can change itself a lot. Now, with SARS, it's an interesting story because this is an infection that first started predominantly in Hong Kong and Beijing and Guangdong province in China, but very rapidly appeared in Toronto. And what happened there is that we had a highly infectious agent that exemplifies this global village we live in. There was a scientific meeting in Hong Kong, someone got sick, they wound up in Toronto, and it was all over the place. But SARS is an example in which we were much better at identifying the agent rapidly by sequencing. We were also able to realize that the only thing that would be effective was quarantine. And this is interesting because, in fact, Singapore was very effective in quarantine. They said, this is what we have to do to stop this, whereas Hong Kong and Toronto were not. Ultimately, it stopped. The dealing with this was very effective. And actually, there were very few deaths if you look at it in a global way. But the effect on the economy was enormous. And so this tells us that even a minor outbreak is going to have severe economic implications globally. And what it did do, though, was help the World Health Organization build a network of reporting, of understanding, of diagnosing outbreaks of diseases everywhere in the world so that we would know how to respond and deal rapidly with them. Now, what I'm going to do is end this talk with a discussion of something that's facing us all right now, and that's Asian bird flu H5N1. This causes influenza. Influenza is with us all the time, various different kinds of strains. This is a particularly frightening one. However, there is no strong evidence as yet of human-to-human transmission. Right now, this is a disease of birds, local fowl, poultry, wild birds. Our concern is that H5N1, which mutates rapidly, will ultimately go from person to person. Now, let me tell you a little bit about this virus because it's relevant. Each virus has a single strand of RNA containing 8 genes. Each gene encodes a single protein. This very high mutagenicity rate, in other words, changing the kind of protein that's made from each gene, can happen by reassorting the genes, by single base mutations, and it just changes rapidly. That's why we get new flu shots every year. And basically, at this time, we know that the transmission of H5N1 goes from ducks to either wild birds or to some cats, tigers, lions, leopards, house pets, with a fairly easy transmission. However, from wild birds to humans does occur. It's not easy. You need very personal contact. And humans to humans, there's not strong evidence for that yet. Our concern is that it might happen. And so what does H5N1 mean anyway? H stands for hemagglutinin, and that is a protein that sits on top of that cage that the RNA sits into. The function of that hemagglutinin is to allow the virus to bind to the host cell and allow entry of the RNA to do its bad stuff. N stands for neuraminidase. Neuraminidase is another kind of protein that's also sitting on the surface of the cell, and it allows newly formed viruses to escape and infect other cells. We have two antivirals out there now. One is called Tamiflu, the other is Relenza, and the neuraminidase is the target for both of these. And in fact, the best way to use something like Tamiflu is if you've not been infected yet, it will give you 80% protection for a while. If you've been infected, it will drop the viral load so that you're not as contagious. You'll still get sick, but you won't be as sick. Now, if we look at the history of flu viruses, the most serious flu pandemic occurred in 1918, and that influenza was H1N1. Killed 40 million people worldwide, and H1N1 means a particular derivative of the hemagglutinin and the neuraminidase. 1957 flu was H2N2, killed about 2 million people. 1968 H3N2 killed about 1 million. I know that one very well because I caught that one. And let me tell you, a real flu infection is no fun. Their current Asian bird flu is H5N1, as I've said. The scary thing about this guy is that right now it has a 50% kill rate, which is enormous. And humans have no immunity against H5, whereas we have some against H1, H2, and H3, which has been around for a while. So what needs to be done? How are we going to deal with this? Let me just tell you first that using something like Tamiflu is best done, in my opinion, not by sprinkling Tamiflu amongst the 30 million people in the world, but rather using it where there's a hot spot. Now that we have an entire network of reporting coming all over the world, keeping their eyes out for hotspots of sudden breakout of H5N1, possibly being passed from human to human, then our Tamiflu has to get there immediately. And then what you do is you cordon off the area, quarantine, treat with Tamiflu, and start vaccines. Now, clearly, the vaccine we have now is to the H5N1 that only goes between birds and possibly cats. What we will ultimately need, if this happens, if it mutates to human, is that we then have to get a new vaccine that will be against that particular variant. And a lot of work is going on right now by many small companies and many large pharmaceutical houses to be ready to make this as quickly as possible. Now, one thing that is important to realize is that vaccines are made in eggs. I mean, zillions of eggs. If you go to one of these vaccine production places, it's astonishing. It's like a football field of eggs. And virus gets injected into each one of these eggs, and then a high titer of more virus is made that's impaired. You kill it, you then make the vaccine. The reason we use eggs is you get a very high titer. One thing that I can't stress too strongly is that, in fact, you can't get flu from a flu shot. It's dead. But you certainly can get immunity. But people seem to think that vaccines are an absolute panacea. It's not true. Flu vaccines are 70 to 90% effective in young, healthy people, and only 40 to 60% effective in people over 65. So that the flu alone is not going to save us. But there are many things that we can do to help ourselves. One of the things we have to do is we have to stockpile face masks, kind you buy in the hardware store for painters, syringes, medical supplies, food and water. Currently, we do not have in the United States enough ventilators if we would have a pandemic. So it's extremely important that we learn how to deal with large amounts of people becoming ill. So what if we do get a pandemic? What does a country do? And what I'm going to do is end with sort of an economic part. And that is, in ordinary times, economic logic does not dictate pandemic preparedness. We all keep low inventories. We don't want redundancies and reserves. We have lots of offshore drug production because it's cheaper. We don't guarantee the purchase of flu drug as we do with other kinds of weapons. In fact, we have just-in-time delivery with no surge capacity. Now, what does this mean if you have a pandemic? The supply chain is very thin. Every hospital contains only 30 days of drugs. We have, in a pandemic, workers becoming ill, drug company workers. The production of new vaccines and new drugs will become less. And in fact, we'll have borders closed and embargoes. They can't get in. Truck drivers get sick. Things can't be delivered. We then have to say, all right, what do we need to do to deal with this? We need scaled up manufacturing and stockpiling of vaccines and antiviral drugs, as well as antibiotics, because many people die of flu infection by a secondary bacterial infection. So we need antibiotics. There's a pneumococcal vaccine. That is something people should all have. That helps. We need better surveillance and epidemiology on a global scale, very accurate reporting of case clusters. We need actual procedures for drug delivery. And most important, we need quarantine laws, not only here in the United States, but everywhere in the world. And our population has to understand what these population laws, what these quarantine laws are before we're faced with the absolute disaster of a pandemic. And you have to know what you're supposed to do in a quarantine, where you'll get your medicines, who will see you. And this can't be done just at a national level. It has to be done in cities and towns where groups of people can work together. That's probably the strongest thing. Now let me just end by turning this back to what are the things that needs to be done with this emerging infectious diseases, with the way in which our world has changed, and the basic science that's going on. So we have now a real need to increase basic research to understand these viral and bacterial pathogens. We have to identify genes essential for the pathogen's survival. We have to sequence and compare bacterial and viral genomes. We have to identify virulence factors and resistance genes and understand how they work. And as I said in my previous lecture, by understanding how the bacterial cell carries out all of its functions to let them grow and divide, we have identified new targets and designed new antibiotics. That's just one lab, and this has to happen in many, many more. The second thing we have to do is design and stockpile new vaccine strategies and in fact make combination antibiotics where you have a particular drug that kills the bug, but in that same pill or shot, you've got a compound that prevents the resistance from being expressed. And then finally, for epidemic control, we have to develop techniques for very fast, hours, not days, not like what happened with West Nile, or even the slowness of SARS, which was much better, to identify causative agents. To do this, we have to exploit viral and bacterial DNA sequence-based technologies. We need, as I keep saying, an increased network of surveillance and reporting protocols, and finally, return to the historical use of quarantine. If we all work together, and if we realize that we are not just independent islands and separate countries, we work together as a global village and help us combat these things. So with that, I'd like to thank you very much.